So I didn't record that much. So yeah, so from here actually we can start. So it's recording now. Yeah. So what I was telling you that we had done those things in the last class, right? So now today what we will do is that we will be going through some of these questions, and also uh, we will actually try to look into the X-ray crystallography field. Okay. So the first thing is that the first question here is the what is the role of alkaline phosphatase in cloning? So if you remember, we talked about molecular cloning, where I have discussed with you about the source of cloning, how eukaryotic proteins, or how eukaryotic protein whose gene of interest, which is a eukaryotic construct that can be cloned into a prokaryote, like in a bacteria. So with the help of vectors, I have said you, with the help of restriction enzymes and also the DNA ligase. Okay, but we missed one thing, that sometimes what happens is that we actually face the problem of self ligation. Means when I have discussed with you about cloning, I said you that you should cut the vector with your you should make a double digestion of the vector with two respective restriction enzymes, and you should use that two restriction enzymes to cut your insert as well. Okay, but what will happen then is that sometimes this restriction enzyme does not completely digest your vector. Sometimes there can be a single digestion of your vector, due to which your vector can religate with one another. Okay, so if that is the scenario, then what will happen is that if the vector is religating, then problem will be is the colonies that you will get in your plate, that they can be your self ligated product, so that is a false positive colony. Okay, so that can waste your time. So what people have done is that they can use this alkaline phosphatase enzyme to minimize this action of this self ligation. Okay, so how to do that? So once you actually get restriction digest your vector, then what it happens is that there is a free 3' OH group and a free 5' phosphate group at the ends of your vector. Okay, so I'm not talking about the insert, because insert is very small in size. So the probability of religating of the insert is much lesser as compared to the vector. So vector is a very large construct and it can actually religate. Okay, so for to avoid that, what people have done is that if you treat this restriction digested vector with a phosphatase, like the alkaline phosphatase. So what is a phosphatase? Phosphatase means to remove a phosphate group. Okay, so this phosphatase means to remove phosphate, or if it is kinase, then it will add a phosphate group. Okay, so here we are treating with alkaline phosphatase. What it will do is that it will remove this phosphate group from the 5' end, and it will make it OH. Okay, so now it is not favorable for this vector to self ligate with one another, because this for this phosphodiester bond to form, we require this phosphate and this OH to do this religation. Okay, but since now you have removed the phosphate group from this 5' end, there is a OH group now, so you will not this vector will not self ligate. Now what will happen is that you have a DNA insert, okay, cut with a restriction enzyme. So in the DNA insert also you will have a phosphate group. So now in the vector you do not have this phosphate group, but in the insert you have this phosphate group in the 5' region. Now what will happen is when you are ligating it with the help of this ligase, now what will happen is that it will actually form this phosphodiester bond. Okay, because there is no phosphate in the vector itself, but there is this phosphate in the insert that is coming from this insert. because you have not treated the insert with this alkaline phosphatase, only the restriction digested vector with alkaline phosphatase. Then what will happen? You see there are two nicks that are being still created. Okay, so you treat it with the ligase, but there are two nicks which are still there. Then you will actually transform this vector into a bacteria. The bacteria has this machinery to refill these nicks. Means they can actually have a particular machinery known as nick translation, where they can actually refill these nicks and make it a complete circularized supercoiled plasmid. Okay, so if you use this strategy, then you are actually avoiding the self ligation of your uh, vector, and you can get actually true colonies instead of false positive colonies. Okay, so many people actually uses this strategy for the ligation purpose. Okay, so this is the use of this alkaline phosphatase in cloning. So then I said you that we will discuss the rest of the questions that I have showed you after we discuss few more slides about what are the different, what is a crystal. Okay, so if you remember that in the last class I mentioned you that the crystal is a array of repeating lattice points, okay, or a array of, someone wants to ask something. Hello? Yeah, Francie, you can ask. Yeah. Yes. Hmm. Yeah. Yeah. Yeah, but yes, that is a valid point, Fancy. But what will happen is that if you remember my last class, what I said that how will you screen your colonies? You can screen them by transforming into a bacteria. Then you will grow that bacteria in an antibiotic plate, right? With the help of an antibiotic selection marker, right? So if you do that, then only those means plasmids which have this antibiotic resistance gene, that complete plasmid means those plasmids, those bacteria which has taken that complete plasmid having that antibiotic resistance gene, those can only survive in that plate, right? But if your insert self ligate with one another, your insert do not have that antibiotic resistance gene, right? So then that self ligation of your insert, first thing is that this insert is very smaller in size, so the probability of getting self ligated of the insert is much lesser as compared to vector. But if you are telling that one insert will sorry, one insert will ligate with another insert, right? In that case it will not have that antibiotic resistance gene, then that will not give a colony in your plate which contains this ampicillin based vector or ampicillin or antibiotic based plate, right? In if that type of insert is been taken by the bacteria, not the self ligated vector, that bacteria cannot survive because it does not have that resistance gene in present in it, right? But if your vector is self ligating, then what will happen is it has this antibiotic resistance gene, so that bacteria can survive and that can give you false positive colonies. So this whole strategies is for minimizing the false positive colonies, right? So now you understand, got my point? Okay. Okay, friends. So yeah, so that is the reason why the self ligation of insert is not that much problematic, but self ligation of your vector can cost you a lot of time, energy, and also your resource. Okay, so now let us look back. What we were discussing is that crystals, right? In the last class, where we means we I give you an introduction about crystals. That I said you that means when actually you diffract a single molecule, and when you diffract a crystal, in a crystal there is a periodicity, means those molecules are arranged in a three-dimensional space in a pattern. and that pattern is repeating in x, y, and z direction, right? So what is that pattern scientifically called? We will define it today, okay? But now you imagine that there is a pattern which is a means more that pattern is actually repeating itself in x, y, and z direction and making a three-dimensional ordered crystal. So if a single thing is repeating it in a xyz direction, then if you diffract that crystal, because of this periodicity you will get a high intensity X-ray diffraction pattern. But if you have a single molecule and if you try to diffract it, the diffraction will scatter and you will not get electron densities. So ultimate aim of X-ray crystallography is to get electron densities of your crystal, and those electron densities we will solve by use of certain softwares and by certain mathematical calculation, and try to determine the coordinates of each and every atom in your crystal. So so we that's that that is how I define the crystal to you. Okay, so but I left where how to produce these crystals. Now you have actually expressed your protein in your lab, over expressed your protein, you have purified your protein, you have a very pure protein, you have checked by SDS space gel electrophoresis, okay? But now for X-ray crystallography, the first requirement is that you need to form crystals of your protein. If you can't do that, you can't proceed with X-ray crystallography. So for that, there are different procedures. One is this vapor diffusion procedure, which is a common procedure in producing the crystal. So in practically, this is theory, but practically what we do is that we actually screen the different conditions of formation of a crystal. Means several protein A will have a separate condition at which it will form a three-dimensional crystal, and protein B will have a separate precipitant, separate condition in which it will form a crystal. Okay, so first what we do is molecular dimension. So there are different screening kits. We use those screening kits to see in which conditions my protein is giving three-dimensional crystals. Okay, but how to prepare the crystal? So there are two different methods. So this is called as vapor diffusion. You can actually learn from the name itself what it is telling. You have some vapors which is actually getting diffusing from one media to another media. Okay, so diffusion is from higher concentration to its lower concentration, something will move. Okay, so that is the diffusion, right, through a medium. So what is here is that there are two types of this vapor diffusion method. One is your hanging drop method, another one is your sitting drop method. So what is happening is that you give, so if you see, first let us understand the hanging drop method. So in the hanging drop method you have a cover slip and you have a reservoir. So this reservoir has a precipitant solution, but that is very much diluted, means that is that has a means this precipitant solution is actually means it has a lot of precipitant solution, right, in this reservoir. But whereas in your protein, in this protein you have actually lesser amount of water, means this drop that you see in the cover slip, that is your protein with little bit precipitant solution, and also the water content here is actually very high. The precipitant concentration in this drop is less, but in the precipitant concentration of this reservoir is much more, right? So what will happen is that since water here is more, precipitant solution here is less, right? But precipitant here is actually less, precipitant solution here is more. In the reservoir precipitant is more, in your protein drop precipitant is less. In your reservoir the water content here, it is actually less because the precipitant is maximum here. In the protein you have a lot of water. So what water will try to do is it will come from your its higher region, higher concentration to the lower concentration, and the precipitant will try to move to your protein. So for forming a crystal, you need to use certain precipitant, and that precipitant normally is 30% PEG, NaCl, sodium acetate. So what it will do, it will actually go and will try to precipitate your protein, but not to that level where you will get powder. It will actually, the crystallization is an art. Means you need to not completely precipitate your solution, precipitate your protein. It should be in a in between zone. So there are different zones of this precipitation. We will discuss in the next slide. So what I am telling you is that there is this exchange of this water and precipitant that will actually try to precipitate your protein. Since this drop is actually hanging in this cover slip, means in this cover slip, so that's why this is known as hanging drop method. But if you are putting it inside this reservoir in a, there is not this cover slip, this is open, and you are putting it as a drop inside the reservoir in a container. Okay, so this is a clear tape there is no cover slip, there is a clear tape in the above. So then that is known as the sitting drop method. So you here you see 2 microliter protein plus 2 microliter of your actually precipitant. Okay, so in this way the precipitant will exchange with water of your protein and try to make a crystal. So this is that you can ask me, sir, then how can we tell that it will form a crystal? So that question is actually people require years to form crystal, or you can get crystals in months. Okay, so that depends upon your protein as well as your screening conditions, as well as the protein formation of a crystal is also a art. Okay, so people, different peoples can do this crystallization at different periods of time, right? So this is the overall method of this vapor diffusion procedure. Now as I said that we don't require, hello, yeah, yeah, so that's why I said you that so the precipitant that I actually showed you, so this is a generalized precipitant that you have, right? But that is a trial and error method. Means first you need to try with certain precipitant. Now you need to see, means in the next slide you will see that there are different zones. Means here if you see there are different zones of the precipitation. So this is known as the phase diagram, right? So what we want is that we want our solution in which the protein is present to be super saturated, right? Then they can form crystals. Super saturated means it should be dehydrated a little bit, means the water should go, then only it will come from a liquid to a crystal phase, right? So that precipitant that you have said that it can interfere with your protein, so you need to screen for that precipitant, that what precipitant will be good for your protein, to so that your protein overall nature will not get hampered by that precipitant, right? And there are different concentrations of this precipitant that you need to optimize before getting the crystal. That's why I'm telling you that what I have said you in one slide, that is a work of 1 year or 6 months, continuous work, that you are doing it, you are not getting, you are trying another method, you are trying to get a crystal. So first thing is that there are different screening kits, so the available in the market, from which you can buy this means screening kits, and you try to screen the crystals condition, mean in which condition you will get a crystal, right? So that you need to actually try start to means it's a game of trial and error. Means you will try and you will get an error, and again you will restart. Okay, so that is the way only that we have now. But then you can tell me that, sir, why we will go for X-ray crystallography? So now this question is very much pertinent, but the thing is that the resolution at which the extracrystallography can give you the structure, means structural details, those that resolution is very much fine for extracrystallography if you compare with NMR, cryo-EM. All of them has some limitations. But in extracrystallography, if you get one crystal, a proper crystal, then you will get higher resolution structures, right? But apart from the alpha fold, I am not talking about the alpha fold, because now people can tell that, sir, we can take a amino acid sequence and directly get the structure from the alpha fold. That is true, but the experimental techniques as well as the computational techniques, if they coincide with one another, then only you can validate that whether the structure that it is predicting, that is actually correct, that is actually present in your body. Okay, so for that, this pain you need to bear, right? Therefore that I am explaining you this solubility curve, right? So at up, means under, when the when your protein solution, that is this drop that you see, this is not super saturated, this is under saturation, you will not get any crystal, right? But as the precipitant concentration increases in that means drop, and your water is going out from that drop to the reservoir, it will go to certain, means these are theoretical zones, but you can imagine it with the help of concentration, with the help of increasing concentration of precipitant. So when it is going, the precipitant concentration is increasing, there will be a point of saturation, okay, where your crystals will be formed or crystals will grow, okay, but nuclei form only infinitely slowly, means your crystals will try to grow, but it will be not a particular, means the crystals actually grow from one crystal to another, means it's not a biological cell, but you can imagine that formation of a crystal or formation of your house. For example, you imagine your room. So for forming your room, you need first a architecture, means on the basis of which your room will be formed, right? You need to first you define with some bricks, which is your nucleation center. For example, that with these bricks in this pattern, this whole room will be formed, means you are forming first some pattern, and the carpenter is following that pattern, and that is filling the blocks and forming your room. So that is the way the crystals actually form. It actually requires a nucleation center where your crystals will start to form, means it is getting some structure, from where if you increase the precipitant concentration, there will be a zone where this crystal will actually grow in larger size, okay, and you will get large size crystals, and a lot of crystals you will get. So metastable zone is that zone where crystals will grow, but the nuclei, means I am telling you, the size of the crystals will not be infinite, or the size of the crystals will not that much larger. Okay, so this is the metastable zones. So in the metastable zone, so there is two things. One is this, in this metastable zone, you can go to the nucleation zone where you will get a lot of crystals. Okay, but in the metastable zone also, if it is not getting that particular nucleation center where these crystals will grow, then the crystallization can stop. So what then people do is that sometimes they used to give small microcrystals, okay, in the protein solution itself. So that microcrystals that you give, they are, as if you remember, like PCR. So there is a template on the basis of which your PCR product comes. So your crystals, protein crystals, will actually be formed with the help of those microcrystals. So that is known as seeding. Okay, so sometimes we can seed at that particular concentration of precipitant with certain microcrystals, which will actually help your interest of protein crystals to grow. Okay, And in nucleation, your complete crystallization formation occurs. So if the metastable, from the metastable zone, it is going to the nucleation zone, so that is the region where you will get your complete crystal. But now, if you start increasing the concentration of precipitate more and more and more, now what will happen? The crystals that are going to be formed due to this nucleation zone and this nuclei being formed for the crystals, those crystals will now be, it will start to precipitate. You will get powder, not the three-dimensional crystal. Okay, so that is known as the precipitation zone. So there is always a competition between this metastable zone and the precipitation zone. In between, you actually get your three-dimensional crystal. So that should be a fine concentration of precipitant that will actually allow that crystal to grow in this nucleation zone, that will allow the nuclei to form, and that will actually help your crystals to grow, the protein crystals to grow. It should not be less, the precipitant concentration should not be less, should not be that much more. If it is more, then it will actually take it to the precipitation zone where your crystals will be precipitated, and if it is lesser, then it will be in the solution, it will not form the crystal. So there is always this competition going on, and in between this there is this art of crystallography. Okay, so this is the protein crystallization in nutshell. So if you want to learn more about this, I will suggest you to go to sir slide and look into its lectures, because I have a little bit amount of time and I need to discuss with you a whole week's content, so I am giving you the summary. Okay. Then what I was telling you, that in the crystals you have certain patterns, and that pattern is actually repeating in the x, y, and z direction to form that three-dimensional crystal, right? So that is how I define crystal in the beginning of the class. Then you should imagine that what is that pattern, what is that pattern, can we actually get hold of that pattern? Because while solving the crystal, or while solving means, if you are going to see Taj Mahal, then you, if you need to build one Taj Mahal like that, means I'm just giving an example. So if you want to build something from scratch, you need to know how the different bricks of that monument is being set with one another to form that whole monument. Then only you can make a new monument, right? So for getting the structural information, you need to first understand the intrinsic part of those crystals. So that intrinsic part of that crystal, or you can say the unit of that crystal, is known as the unit cell, which actually repeat by, if you translate that unit cell in x, y, and z direction, you will get a three-dimensional crystal like this. Okay, so in this slide you will learn about, means you will see these terms, crystal lattice. So I will discuss with you what is a lattice, what are these different points, what are these different coordinates which actually define your crystal, and this information is very important to understand to finally get into the image of your crystal. Okay, so once you have done, have all this information, then only you can construct the image of your crystal and get the structure, right? So this diffraction intensity means, what I have said you, that this x-rays will come, this is your crystal, it will hit this crystal and diffract, this x-rays will diffract, and you will collect the diffraction in a detector. And the diffraction pattern or the diffraction intensity means, all these points that you get, these points you will get as electron densities in the detector. That electron density's intensity, that is the diffraction intensity, is actually proportional to the number of unit cells in the crystal. So more the number of unit cells, more is the intensity. Why that is so? Because the telling you electron density. Electron density means the electrons are going and hitting that detector, and you are getting the intensity of that electron. So more number of electrons means more intensity, more unit cell means more number of atoms or which are being arranged in that unit cell, and your crystal size is also more, so you will get larger intensity in the diffraction pattern, right? So so more the number of unit cells, larger is the intensity, diffraction intensity. Okay, now there is a concept that now you can ask me that, sir, that you have said that in physics we have learned that these waves, they have a constructive nature as well as destructive nature. So the x-rays that are going through this crystal, so if we can get a crest in the electron density pattern where in the detector where these waves are going and striking, because electron has dual nature, it has a wave as well as particle nature. So when it is going and hitting your detector, so then it can be a constructive interference between two waves, because multiple waves will come, right? So electrons from multiple atoms are coming and interfering with one another, that is known as interference. So this interference can be constructive, means both crest, means what, how a wave looks like. So a waves look like this, right? So this is your crest, right? And this is your trough. So if two waves are like this, so there is this is this, and there is this, and then there is coming this. So this is wave number 1, this is wave number 2. Now, if you get an information from adding up these two waves, then both of them will cancel out one another, because here what is straw, here it is also a crest, right? So this straw will cancel out this crest, because this increase and decrease, if you add them together, it will subtract with one another and it will give you 0. So that is a destructive interference. To get a point like this, you should be having the waves of your light, the x-ray, here it is x-ray, in falling in your detector in a constructive interference. So the wave should come like this. So this should be one wave, this should be one wave, okay, and this should be one wave. So here all the crests are adding together to form a greater intensity or giving you a particular point like this. So how then you can ask me that, sir, how we can actually do that? Means how we can decide whether it is a constructive or it is a destructive interference? Means whether we will be getting any electron density or not, that depends upon whether it is a constructive or a destructive interference. So that is actually explored by various physicists, and they have actually worked on it, and there are different mathematical inference that if you actually use a particular, means if your crystals are in a particular distance, means these all these lines, if I draw one line from here, corresponding all these points, this will give you a plane, right? Means if you have a line, this is a line, and if you have these two lines in two axes, then what you get, this is a plane. So always imagine that we are talking about something which is a three-dimensional form, and I am drawing in a two-dimensional sheet. So always imagine that when I am talking about something, if I don't mention, there is a axis which is coming towards you, right? So there are two axes if you see, and these are all planes. There is a distance between two planes which is known as the interplanar distance, that is this d. And you will see that on the basis of this d and the wavelength of your light, these two actually decides whether the wave that is going, that is diffracting from these atoms, it will be a constructive or a destructive interference, and that is given by the famous Bragg's law, that is 2d sine theta equals to n lambda. So I am just leaving it here, but we will coming to this Bragg's law in the later slides. Okay. Now, what is very important here is to look into the thermodynamics of the crystal formation. So, what happens is that, so if you remember, when in the search lectures as well as when we discussed about the protein folding process. So in the protein folding process, if you remember, we have talked about that this enthalpy there is this moderately, this is actually the enthalpy is unfavorable, right? But due to the entropy of the water molecules, you are actually getting the protein is actually folding, because if you remember, del G equals to del H minus T delta S. So when a protein is folding, it is going from its linear form to a structured form. Means it is losing a lot of entropy when it is going from a linear form to a three-dimensional structured form. And this huge loss of entropy should be thermodynamically unfavorable. But then how we are getting the protein in this three-dimensional form? This is because the water molecules which are present in the means in the surrounding of your hydrophobic amino acids, those water molecules will be now free once those hydrophobic amino acids are together. Because of the hydrophobic collapse of your linear polypeptide chain into a three-dimensional structure, it gives that del G value to be favorable for the protein folding, right? So this actually so that is that concept we are actually again coming for this crystallization, because in crystallization also your proteins are going to a rigid crystal. Means the dynamic nature of your protein is actually compromised, right? Means if your protein has certain loops, the one thing is that if your protein has more unstructured regions, like you have long random coils or loops, not secondary structures, then that is very difficult to crystallize, because those regions will be actually creating trouble for the packing of your crystal, and also making the whole process of crystallization thermodynamically unfavorable. Because itself, when you are putting a protein into a crystal, that entropy of that protein is getting reduced, because you are putting it is like the all those means like ships are running in your field, and you need to bring all those ships into a house where you need to keep those ships together. Then you are actually uh, keeping the means the ships were running here and there. Now you are putting all those ships in one house, so the entropy of the or the movement of the protein is reduced. So the entropy of your protein is unfavorable always. But since you are doing this, you are actually putting all this sheep in a house, you are actually giving the grass to survive. Means I am giving an analogy. Means the sheep would have eaten a lot of grass, but now the grass are present in the open field, and it can actually, when air is coming, it can move to and it is happy, right? So similar thing is happening with your solvent. When your proteins are coming and together into a fixed structure, the solvent is now actually free to move, like we actually learned for the protein folding process. That's why your delta S solvent is actually largely favorable, and this large favorable is actually masking the unfavorable entropy of your protein, and making this term, this entropic term of means largely means making this means a term negative. Okay, because this is much larger, so this unfavorable is less unfavorable as compared to this large favorable entropy of a solvent, and making it little bit negative. Now what is happening, that if you imagine what is happening, that is given here, means delta G crystal. The entropy here is small favorable because of this hydrogen bonding. Means when a protein is coming in a crystal, it needs to interact with one another as well to form actually a three-dimensional structure, and there are different hydrogen bonding which is happening in that process. So that hydrogen bonding is actually increasing your enthalpy in us, means increasing your enthalpy, and if it is a very large increase, then the del G will not be negative, right? Because if it is more, if this delta H is more, enthalpy is more than this entropy, then this delta G will be negative. So the delta G will be positive. So that is unfavorable, right? So that's why this delta H is favorable, but very smallly favorable, because the hydrogen bonding will occur, but this is not like a secondary structure making hydrogen bond. It is a complete three-dimensional protein which is interacting with one another, right? So there is a small enthalpy, the enthalpy is modestly favorable for a crystal formation. The entropy of your protein is largely unfavorable, but the entropy of your solvent is again largely favorable. So it is minus small plus large minus large. So these two are negative, right? Means this delta H is also negative because it is little bit modestly favorable, and this solvent is largely favorable. So both of them are negative, but entropy of your protein is unfavorable, that is plus large. So overall the del G value is equals to less minus small, means from here also you can see there is a very small possibility that you will get a crystal. Thermodynamically you can also get it, that there is a very small possibility in that region, in that particular temperature, in that particular solvent condition, you are going to get a crystal. So this is the thermodynamics of crystal formation. Now how you can actually, this is a small, means small negative del G value, means this is favorable but not that much favorable, this is moderately favorable of the crystal formation. So to increase the chance of formation of crystals, what people have done is that first you need to minimize the floppy ends. Like for example, if some protein is like this and it has a C terminal end like this, so what people do for crystallization, you will see that they will chop off this part. So for this, you are actually removing the information, some information you are losing in this part. But if you don't do that, you will not get the crystal, because this is a very disordered part. So to then you can tell, sir, then how we can actually we are losing some information. For that you have NMR. So if your protein is smaller and you have various these loop regions like this, okay, and there is some structured region, then again some loop region. So then this protein structure can be easily visualized, and also the dynamic nature of these loops can be visualized by NMR. But for X-ray crystallography, what people do is that these floppy ends are actually cut with the help of certain enzymes. Okay, and you will have some regions which are already structured. So that will actually help for the crystal formation. Then if you have multiple domains, then what people do, express individual domains separately and then crystallize them separately. Means there can be the problem that this is one domain, then this is another domain, and in between them you have this long loop region. So now you, if you cut this loop region, you are missing this domain. Then what to do? Then you take this domain individually, clone it, express it, and form one crystal. Again, you take this domain and try to form another crystal. So you are getting information about how these two domains look like by two different X-ray crystallography experiment. So that is the strategy. Sometimes what happen is that some proteins, like some transcription factors, they are like this random coil form. Okay, much of their part is random coil. When you give a substrate like DNA, this random coil will now form a structure like this. Okay, so this original structure is present, like this domain was present, but this random coil is now forming a ordered structure when you add a ligand. So that can actually make your overall structure compact or overall structure suitable for the crystallography. So sometimes we actually add ligand which can actually stabilize the structure of a protein. Or sometimes what we do is that if you have some high entropic residues, like you have glycines which are mostly present in these floppy ends. As I said, that glycines have high entropy, and glycines and proline, they want to stay in these floppy ends only, or in the loop regions, right? So if you try to mutate them with some amino acids which will not hamper the overall structure of your protein, but it will reduce the entropy of your side chains. Okay, so then you can, that is also one strategy many people use for making this small del G a large favorable process for crystal formation. Okay, so now so how actually uh, we can differentiate between, so now as I said you that in your precipitant there is salt, there are salts present as well as your protein. When you are actually precipitate, when you are actually trying to form the crystals, what is the surety that you will get only the protein crystals? You may also get the crystals of your salt, right? Then how to actually separate that whether this is a protein crystal or this is a salt crystal? Because this crystallization process is a very huge time consuming as well as money taking process, right? If you are taking a salt crystal and try to have a X-ray diffraction pattern, you are wasting money, you are wasting your time. Okay, so what people do is that they have different strategies through which you can actually see whether your crystal is a salt crystal or it is a protein crystal. So there are different kinds of dyes which can actually specifically interact with your protein. So if you get those dyes into that crystal, then that, because these crystals have so what is important to know is that if you, I am telling you the water is moving out from the crystal, then only it will form crystals, right? But protein is a biological sample. If you too much dehydrate your in the formation of the crystal, then the protein may precipitate, because these proteins are, then the crystal will not be formed. So for biological samples, what happens is that 70 to 80% inside that crystal you have actually water molecules present. Okay, so there is always a balance between the dehydration procedure and the amount of water that is required for your biological molecules to keep intact. So that's why while crystallization, what we do is that we actually freeze those crystals in a minus, means in new in liquid nitrogen. We actually do a process known as flash freezing. Okay, so in the flash freezing, what we are doing is that the water molecules which are present in the outside of your crystal, those water molecules will only get into the form of ice, or they will be dehydrated, because you are freezing the crystal in a very short period of time. Inside your crystals there will be still water channels present. Okay, so that will keep your protein to be in a hydrated form and not allow the biological function of the protein to be lost completely, right? So for that, if you are actually taking a protein crystal and a salt crystal, in a salt crystals you will not have these water channels, so they are much more brittle. If you try to break that salt crystals, it will be breaking with a crack. Okay, but if you are breaking a protein crystals, they are much more softer than these salt crystals. Okay, so they will be broken very easily, the protein crystals. Okay, sometimes what you can do, you can perform dehydration of your crystals and try to see if it is if you are dehydrating a protein crystals, since it has much of the region, it has solvent, everything will be dehydrated or the dehydration will be faster. But when you have salt crystals, it does not have that water content that much, that if you dehydrate it, you will find those salt remnants present always. And the simple way to look into it is that not the simple, it requires a in-house X-ray crystallography source. If you have one, then you can actually take that in-house X-ray crystallography source and diffract your crystals and and try to see that whether they are forming a three-dimensional pattern or not. So from initial X-ray diffraction, you can tell whether it is a protein crystal or not. So that's why when you are preparing one crystal, it is actually very helpful if you have a in-house X-ray diffraction source. Okay, so once you have made a crystal, there is a cryo loop. As I said, you do this flash freezing in a actually very low temperature in liquid nitrogen. So there is a loop with which you need to handle these crystals. You are seeing these images like beautiful images like this, but those crystals that will be formed of your protein, you can't even visualize by your naked eye. You need to visualize it under some microscope. So from the microscope you are taking a cryo loop, and with the help of there is surface tension, and there is something known as mother liquid, so on in which your crystal will be actually stable, right? So you take some mother liquor and you take that crystal in and mount it in a goniometer. So what you see here, this is a goniometer. So this goniometer, in this top it has this loop which is where your crystal is present, and from here this X-ray beams will be coming and it will diffracting the crystal and it will fall in the detector. But what this goniometer serves the purpose is that if you don't rotate your crystal, then always the X-ray is coming and hitting some atoms and giving you a electron density pattern, but you are not getting the information of your whole crystal, right? So for that you need to create certain angles at which your X-rays will hit. One is that you need to get the whole information about that crystal, and also what you need to do is that you need to actually take care of certain angles in which if you hit the X-rays, as I discussed with you in during the Bragg's equation, that in that angle if it hits, or with that wavelength of X-ray if it hits, then only it will give you a constructive interference. So all these things should be taken care of. For that you need something which is more flexible. So this goniometer head has three angles in three directions. You can actually rotate this loop which is attached to this goniometer head. So one direction is perpendicular to your beam. So if your beam is coming in this direction, so in one angle with which one axis in which you rotate, that is perpendicular to the direction in which your beam is coming. One is perpendicular to your loop axis, means if it is your loop axis, one axis will be here through which you can rotate, and one is a magic angle. This magic angle is actually fixed. So this angle is fixed in a goniometer head, that is your chi angle. So chi angle is fixed, that is 54.74 degree, and other two angles you can actually adjust so that you are getting a constructive interference, and also you are actually diffracting the whole crystal. Okay, so this is the crystallization procedure. So now for your actually means this slide, this is crystallization. Okay, so this is not crystallization, this is crystallization. So so this is just for giving you means in history, means how this different, what is the first crystal, that is that of this pepsin by James Summer. Okay, so and in by, and also the final so the first crystal that was whose structure was been, or first protein that was crystallized was an enzyme known as urease by James Summer, and the crystallization of that means to the structure of that protein we got a pepsin in 1930 by John Northrop. Okay, and the first X-ray diffraction pattern of pepsin was actually collected by Burnell and Crawford. Okay, so they were the students of this scientist, and they and but the first three-dimensional structure that was being solved. Okay, the first three-dimensional structures that we solved, that was of myoglobin by Kendrew. Okay, and he won the Nobel Prize in 1964. So this slide will actually help you to solve some of your questions in assignments as well as in your exam. So that's why I have kept it. So this is some history about protein crystallization. Now we are moving to the X-ray source. Okay, so now what we have discussed till now is that the overview of X-ray crystallography. Now we are digging deeper into it. So we are going that I have said you that X-ray is coming from a source. Now you should have a question in your mind then how the X-rays are produced. Means what is the source from where these X-rays are coming. Right. So we will be discussing that. Before that, I had some questions. So the first one I had already answered in the first hour, first half an hour. Then I said you. Then there is a question. If there is 60 to 80% water inside the crystal, when we cryofreeze the crystal, how come the water inside doesn't crystallize? So this answer I have already given you because we do a flash freezing in very small amount of time we actually freeze our crystals, right? So for that the water which is inside your crystal that is not getting that much time to get crystallized, whereas the waters which are present in the surface of the crystal that molecules will get crystallized, and that due to which you will have 60 to 80% water still available inside your crystal flowing through different channels. Then you should have this question in your mind that if water is inside the crystal, then when we are heating it with X-rays, then what will happen? The protein is also there, then water is also there. Water will also give some electron density. Then what will happen? It will actually be we should we get the electron density of the water as well. So to answer that, what you should remember is that these water molecules are not arranged in a particular three-dimensional form or in a ordered form. As I said last class, as also today, that if you want to have a higher intensity electron density group, then you should have a periodicity. Means you should have something which is repeating itself in space in a particular pattern, then only those electron densities will add up and you will get that kind of spots. If those information is available just like in water, then what will happen, the diffraction that you are getting from water, that will not be coming to a single spot or single electron density, it will scatter more, right? So that will add up to the noise, and to cancel those noise we have various kind of filters, which I will be talking some of them if not today in the next class. Okay, so we have different kinds of filters which will not allow this noise coming from these water molecules to go into your final detector. Okay, okay, so that is the answer to this question. Now, does we get hydrogen ion electron densities? So, why I have asked this? Because this is very important. Whenever you actually download some structure from this PDB, protein data bank, you will not find any hydrogen ions attached, means hydrogen ions information in that PDB structure is not available. Always you will get some proteins, but the hydrogen atom, the whether where it is actually coordinates are present, that is not actually directly available from a X-ray crystallography structure. Why that is so? That is so because I am repeatedly telling you the molecule which will have more number of electrons, that will diffract more amount of X-rays and you will get electron densities for that. Since hydrogen atom has only a single electron, that electron's intensity that will be present in the diffractometer or in the final detector, that is similar to the noise that is present. For that you cannot actually pinpoint the coordinates of the hydrogen atom for of your crystal or in a X-ray crystallography. This is a limitation of X-ray crystallography. Okay, so since I have now answered these questions, Now let me little bit discuss about the how X-rays are being produced. So we will be trying to means cover as much we can, because I don't want to rush a lot, because my basic is that if you can understand at least 10% to 20% what I am telling you today, that will be a gain for me. Not like means ending slides and sites and you don't have any information in your mind. Okay, so we will go to that extent where we can, means to the slides where we have time, because I will also discuss with you about the week 3 assignments. So till means I would say 7:45, to which slides we will be going, that will be the end of today's slides discussion. Then I will discuss with you about the week 3 assignments. Okay, because that is also my duty to discuss with you. Okay, so now we have known about the overview. Okay, so if you guys have any question, you can just stop me in between and you can ask me, right? So that will be good. Means if for that, if I can't complete one slide, that is fine. If I can't answer, that is also fine, because I will read and I will come back next class, just like today I what I did. So so that in that same manner I want to carry out this session. Okay, so if you have any doubt, you can stop me in between. Okay, so we are moving further and we are going to the source of X-ray crystallography. Now, as I said, the first X-rays were being actually if we look back into the history, William Röntgen, actually Röntgen was the first one who actually discovered this X-rays, from which he actually called as the cathode rays tube. Okay, from some cathode rays tube he actually come up with this production of X-rays. So what is this cathode ray tube? So this cathode ray tube actually has, these are two electrodes. One is your anode, one is your cathode, and this is actually present in a single chamber. Okay, where inside this chamber there is vacuum. There is no air present inside this chamber. You have a cathode. You have a cathode which is negatively charged, and you have an anode which is positively charged. Okay, now, how this negative charge is maintained? This is maintained by a high voltage source. Means you have a battery or a high voltage source. With the help of high electric current and voltage, you are creating a negative charge in this electrode and a positive charge in this electrode. In the anode you have positive and in the cathode you have negative. As you increase the voltage more, these electrons which are present here in this cathode, because of the heating effect of this cathode, these electrons will be ejected out from this cathode. And since the electrons are negatively charged, they will try to move to a positive termini, that is your anode. And since you are giving more and more voltage, the velocity of the electrons will increase and it will try to move in a very faster rate towards this anode. Another important thing is that you do not have any air molecule in this sealed tube, due to which you will not have any hindrance. Otherwise, if you have air inside this tube, then those electrons which is being emitted from this cathode, they would have interacted with your air molecules. But here there is no air molecule, there is vacuum. So these electrons will move at a very faster rate and try to hit this anode. So from this anode is actually made up with certain materials or metals like copper or molybdenum. Why that is so? Because you know, if you remember the basics of chemistry, this copper, molybdenum, they are all d transition elements. They have the capability of these d transitions. They have certain empty orbitals in which if you actually try to increase the energy of the electrons from the ground state, that electron can go from the ground state to a excited state in that empty orbitals, right? So can utilize those empty orbitals which are present in this d means transition elements like this copper and molybdenum. So we normally use this copper and molybdenum, not we, means when X-ray is produced, these two are normally used. So then you can ask me that there are so many DD transition elements, why only copper and molybdenum? So wait for it, I will give you the answer. Okay, so when these electrons are hitting your copper anode, what will happen is that your copper will have a nucleus, means the copper atoms will have a nucleus, and they have electrons which are present in this orbits. So two things can happen. So that energy, the electrons that is actually giving into this anode, and this anode will be getting heated up, due to which there will be some radiations which will be emitted from this anode. Okay, so these are the X-rays. So how these radiations can emerge? One is the either these electrons which are present in these orbitals, they can go from their lower energy state to the higher energy state because of the energy of the electrons, or the velocity of the electrons that they have gained from this cathode, or the electrons can directly hit the nucleus. The nucleus is positively charged, right, because of this proton. So the electrons can directly hit this nucleus and will not interact with any electron in the orbital, and can directly diffract out from the copper atom. Two conditions can happen. One is this one, another one is the electrons in the orbital from the ground state, it is going to higher energy state, right, when a moving electrons is hitting into the copper atom or a molybdenum atom. When this is happening, means it is not interacting with any electron, but it is just interacting with the nucleus and going out, then very low intensity X-rays are produced, because there is no discrete energy gap that is being produced, because it is a continuous process. Some electrons will come, they will diffract, they will go through this empty space and they will diffract. This process is known as Bremsstrahlung. Okay, Bremsstrahlung. Okay, this Bremsstrahlung, if you see this curve, this is a famous curve whenever you actually learn about X-ray crystallography, means how these X-rays are produced. You see there is a continuous region here. So this is your this Bremsstrahlung. When this in electrons which are very high energy in electrons, they are not interacting with any electron present in the copper atom. They are going through the empty space through the nucleus, and they are producing X-rays, but they are very low intensity X-rays. But remember, for crystallography we require very high intensity X-rays. For that, when the electrons, this moving electrons is coming and their energy is able to actually move these ground state electrons to their higher energy level, that is from this K level to this L level or M level, these are discrete energy levels, and in each of these energy levels they have some vibrational levels, means these energy levels are also again divided into several vibrational states. Okay, so what two things can happen. So one thing is that from the k orbital it can move to the L orbital. The electrons can move from the K ground state to the L excited state, or it can move to the M excited state, right? Once it is going to the excited state, it will after some time, it can't be present in the excited state. Everything wants to come to a stable form. So what will happen, if when the energy is it is going to the excited state, it will again come down to its ground state, and in that process, when it is coming from the excited state to the ground state, the energy which is actually emitted, that is in the form of X-ray. So what is very important is that you need to take the gap of this orbital in such a way that you need to have a particular, means whenever this energy is coming, so the drop of the energy is proportional to the wavelength of the energy that is emitted. And remember, we when we were talking about that, why X-rays we use, because we use X-rays because it is in this 1.54 angstrom wavelength. and the distance between two atoms in a crystal is in this range only. So I gave you the example that whenever you try to visualize something, that visualization software or that visualization lens should be compatible with the distance in between that object. Means that is how you get a resolution, right? When you are actually distinctly telling that these are two different points. Okay, so the distance between two atoms, that is the carbon carbon single bond, that is 1.54 angstrom. That, and the wavelength of the x-rays with which we are diffracting the crystal, they are also 1.54 angstrom. Then only you will get this x-ray diffraction pattern. So to you will get this 1.54 angstrom only when you actually use this copper anode, because this energy gap is such in copper that you are actually getting this 1.54 angstrom. So this 1.54 angstrom is for this K alpha transition, that is from K to L level. So there are two K alpha 1 and K alpha 2, because in L energy level, the vibrational states are far away from one another. But in M, you have only a single thick line. You see, the vibrational states in M energy level is very, if they are very near to one another. That's why you get only a K beta absorption for the K to M K to M emission, M to K emission. Whereas for L to K emission, you get two, K alpha and K alpha 1 and K alpha 2. For copper, if you these are the K alpha 1 and K alpha 2 values. These are the wavelength values, right? So if you actually try to means average these two values, you will get 1.54 angstrom that we require for to diffract our protein crystals. But for small molecules, we generally use this molybdenum. Since molybdenum K alpha value is 0.7 angstrom. So these 0.7 angstroms, because in small molecule means what? Means all those atoms, the distance between these two, those atoms are very much smaller as compared to large molecules. Then the copper electrode will not serve that purpose. These small molecules are normally used by chemists. Like if they want to try to have the X-ray crystal structure of certain salt complex, then they actually use this molybdenum anode to produce the X-rays. But if you are working with a protein, like in a biology laboratory, then your crystal size is larger. Around the distance between two atoms is around 1.54 angstrom. Then you normally use the copper anode. Okay, so the anode choice is on the basis of how much, what is the dimension of your sample that you want to use for your X-ray crystallography. Okay, but then one question should still remain in your mind, that what will happen to this K beta, because this K beta will also come. Then you will have a mixture of K alpha and K beta coming to your protein, but you don't require this K beta value. So how to remove that? That removal of this K beta wavelength is through again some filters, where you will get, if you pass this X-ray through those filters, you will get only this K alpha wavelength coming out from that filter, and that will actually go and hit your crystal. So what are different types of filters? If I don't have time today, we will discuss in the next class. So this is the X-ray source, the primitive X-ray source, the concept of how X-ray produce. But there are majorly three different X-ray source. Means this logic is being used mostly in forming the primitive this seal tube. Okay, so these seal tubes are actually used in the primitive, means they are not normally used today, because seal tubes have a lot of disadvantage, right? Because the one thing, the first disadvantage of this seal tube is similar to what I have discussed here. You have a fixed anode, and always the cathode electrons are hitting this anode. Now this is not a everlasting anode, right? So it will not like, it is like made up of diamond or anything, that it is always avoiding, it will always stand there and tell that come electrons hit me, right? So it has also a lifetime. So if you have something which is fixed and you are continuously hitting it, sometimes what you will find that anode, some pores will be produced in that anode, and your anode will be not of no use, because now the anode is destroyed. So in a sealed tube, you have a fixed anode. It is not rotating anywhere. It always, the electrons are coming and hitting it in a particular angle, and it is generating heat, huge amount of heat, due to which there will be pores in the anode, and you will get very low intensity X-rays from these sealed tubes. To combat that, so sometimes what people do is that there are certain water channels in this seal tube which can actually lower the temperature of the anode. So these waters will come and go from where this anode is present, not directly interacting with the anode, but there is a cold water chamber which is which is beside the anode, so which will keep this anode colder, but still this anode half life will be very less. So the advantage of the seal tube as compared to the rotating anode is that the vacuum here produced, because this is a seal tube, this is a single tube, so once you have produced the vacuum here, it will be vacuum, because you are not opening anything. Okay, so you are just collecting the X-rays, but this vacuum is intact always. But in the seal tube, your lifetime of your anode is compromised. But in the rotating anode, what here it is happening is, from the name itself you can imagine, your anode is continuously rotating. So what advantage it will give is that always one surface is not accessible to the X-ray beam. When you are rotating the anode, at every angle difference, you are actually at different surface of the anode, these X-ray beams are hitting, due to which it will increase the lifetime of your anode. In seal tube, what it was happening, it is hitting at a single region, due to which there will be pores. But now you are increasing the lifetime of the anode, and you are also reducing the heating of the anode, because they are continuously rotating this anode. So from this rotating anode, you can actually get higher intensity X-rays as compared to the seal tube. But what is disadvantage in this rotating anode? Because of these rotating chambers, there are different chambers which are associated with this rotating anode to rotate this, right? Due to which it is very difficult to maintain this vacuum, and always you need to actually refill, you actually maintain this vacuum continuously after some period of time. When you will see that the X-ray intensities is reducing, then you need to again create vacuum inside this chamber. Another important thing is that this X-ray beam from where it is coming, this X-ray lamp from where it is coming and hitting your anode, from the cathode where it is coming and hitting your anode, it should be adjusted in a particular angle, because this is always rotating, right? So there may be chances that this is coming and not hitting the anode. So you should always have a fine alignment of this X-ray beam from where it is, means the cathode from where it is coming, the electrons, and the rotating anode. So you need to always check that alignment. When that alignment is getting distorted, you need to again bring back that original alignment, so that you can get high intensity X-rays as compared to seal tube. This rotating anode is present in normally the home source. Means what I am telling you, that in the starting when I said you that uh, when you try to differentiate between a protein crystal and a salt crystal, you can use a home source to diffract it with the help of X-ray. So those home sources actually follow this rotating anode, but that will not give you a high resolution structure, because the intensity of the X-rays are still less. So how to produce that huge amount of X-rays in higher intensity in a short period of time? For that we have synchrotron. So these synchrotron are present only in specific countries. In India we don't have. What we do is that we prepare our crystal, we cryofreeze them and send it to a synchrotron facility for capturing our, actually for getting a high resolution images. Okay, so sometimes this in-home source is also means fine, but it cannot give the images that a synchrotron can give. Okay, synchrotron can give much higher resolution images because it can produce in x-rays in much higher intensities. So this synchrotron, if you see, this is a synchrotron. So this is present in France. So there are synchrotron in France, in Germany, in Italy, in Japan. Okay, so there are 3 to 4 countries where you have synchrotron. And as you can see, this is the image of a synchrotron. This is a whole village. So this is a huge tunnel surrounding a whole village. So this is like a whole city which is comprising of this synchrotron. So there is a tunnel that you can see here. So what is this tunnel used for? So this tunnel, in through this tunnel, the electrons are actually moving near to the speed of light. Okay, and they are moving in such a high speed, and every time they are moving in a circular path. If you imagine now that when an electron is coming in that high velocity, so what it can do is that this electron will come and try to hit here before move to here. Why it is moving in a rotating path? The electron should try to there, because of this centrifugal force. When you centrifuge a sample, it is, there is a force always driving it outward, so the electrons will always face that. So to keep this circular motion intact, you have different magnets which will actually deflect these electrons in that angle that these electrons will always be moving inside this tunnel in a circle and circle. But always the electron will try to hit in this wall. But when you are actually deflecting this electron into the circular tunnel, the electron is losing some energy there. That loss of energy is being collected by different windows which are present here. So for X-rays, we know that at what energy or wavelength we require, and that at that window we will open and we will collect the X-ray. So from the synchrotron, you can get different X-rays, other sources as well. So you will open the gate at that particular velocity of the electron and particular energy of the electron in which you require the X-ray. Like for 1.54 angstrom, we know that at what particular velocity of the electrons, mm, we need to open that window to collect the X-rays. But continuously after that is done, again some electrons are circulating in that light speed inside the tunnel, forming a high energy field, high energy magnetic fields which are present. So these high energy magnets, some of them are these bending magnets or wigglers. Okay, that can change the momentum of the electrons. That is actually responsible for the electrons to move in this circular motion. So if you have a synchrotron, then only you can actually, there is no concept of this anode and cathode and the half-life of this anode, anything. So this we are utilizing for high energy X-ray production. So this is the concept of synchrotron. So so this is again the history of some X-ray generation. So as I discussed, Röntgen first produced X-rays, and all these things you can actually go through by yourself. This is only because you need to solve the assignments, and some of these can direct question come as direct question in your exam. Okay, so you can just read about this. Now let me explain you the Bragg's law. Means from here, if you see, William Henry Bragg and his son, they actually got Nobel Prize in physics for actually explaining the diffraction of X-rays by crystals. They theoretically they have done that. Another group of scientists, von Loy, and his students, this von Loy, he is associated with Sommerfeld, and Loy's students Walter Feldrich and Paul Knipping, they together actually theoretically derived that what are the conditions that needs to be satisfied to get a X-ray diffraction pattern. So there are certain conditions known as Law's conditions. If those conditions are satisfied, then only you will get a constructive interference. Okay. Similarly, Bragg solved an equation, gave that Bragg's law, which also point out the same story but in a different way. Right. So first let us understand the Bragg's law. Bragg's law, as I say, that if you imagine these are the two lattice planes of your crystal, these are two planes of your crystals, means they are, so if you imagine this is the whole crystal, okay, if you imagine this is the whole crystal, and so I am, pardon my drawing, but yeah, so if this is your whole crystals, so this is one plane of your crystal and this is one plane of your crystal, and in between them there is an interplanar distance that is denoted by d. He said that, so now imagine when this is your X-ray source, from here the X-rays are coming. Now X-ray will hit this plane and also this plane, right? But the X-ray which is hitting this plane and the X-ray which is hitting this plane, second plane, for the second X-ray beam to hit this second plane, it need to cover an extra path to come out and diffract into the diffractometer, right? So if you compare the beam, this beam with this beam, it need to always cover a extra path, then only these two are similar, from where you can actually tell that these are going and reaching into the diffractor. So from here only you can get this kind of pattern, constructive interference, if both of them are reaching the detector at a same phase, right? But always the X-ray which is hitting the nearby plane, it is covering an extra path. Then what should be the relation of this path difference with the phase difference of this wavelength? That relation is given by, to, so that it can form a constructive interference into the detector. That is given by your Bragg's law in a mathematical form. So if you remember, if you draw a triangle here, this is your D, that is, this one is your D. This is your hypotenuse of this right angle triangle. There is a right angle, there is this theta, which is your semi diffraction angle. Okay. If you take sine theta means perpendicular by hypotenuse, right? Sine theta means that. So the hypotenuse here is this D, and what is your perpendicular? So the perpendicular here is this AB. So you have, if this AB can be written as D sine theta from that. Similarly, BC can be written as D sine theta. If you add together them, then this AB and BC is your path length. And what Bragg said, that to satisfy the constructive interference condition, your path length should be an integral multiple of your wavelength of the light that you are using. The wavelength of the X-ray should be directly proportional to the path length this X-ray needs to cover. What is the path length? Path length is AB plus BC. That is your 2D sine theta, because AB is also D sine theta from this trigonometric calculation, and BC is also D sine theta. So if you add them, that is 2D sine theta. That is equal to, if you actually, I said, Bragg said that this should be proportional to lambda. If you try to open up that proportionality with the proportionality constant, that is this n. Okay, so 2D sine theta is equals to n lambda. So this is the famous Bragg's equation. This n can take 1, 2, 3, any integer value. Okay, any positive integer value. Now you see the sine theta value should be always -1 to +1. It cannot go, the sine theta value is not more than +1. Okay, so to satisfy that, so to maintain that, this n lambda should be always less than this 2d. Then only, because sine theta is what? Sine theta is n lambda divided by 2d. So if this whole value, this sine theta value, if it is less than 1, then this n lambda should be less than 2d. You understand, right? This, you put it here. So this is n lambda divided by 2d, right? So now this sine theta value should be less than 1. So if the sine theta value is less than 1, then your n lambda should be less than 2d. 2d means twice the distance of your intraplanar distance. D is your intraplanar distance. So if n is equals to 1, then your lambda should be less than twice the distance of your intraplanar distance. Now you understand why I am telling you that why we require 1.54 angstrom of your X-ray to get the image, because that 1.54 angstrom is either less or similar to the distance between these two atoms. So that's why we are using 1.54 angstrom only for the diffraction of your crystal, right? So this is Bragg's law. So how much time we have? So this is already 1956. So 7:56 already happened. So from lattice I can continue in the next class. Okay. If I have time, then I will talk today only, because we started little bit late, around 6:15. So we can continue this class to 8:15. But first let me discuss with you the week 3 assignments. Okay, because that I need to first discuss with you. Then only, if we get much more time, then I will discuss with you further. And also, if you actually see that this means I had made some practice questions for you, but I didn't get much time to for solving these questions today. I will share these slides with you. Don't solve the all the questions, because everything I will not be covering today. So better I will solve these practice questions in the next session when many people will be here. I don't know why people are less today, but yeah. So let me discuss with you the week 3 assignments first. Okay, week 3. These are the week 3 assignments, not week 4. Okay, so I think you have already submitted the week 3 assignments. So I will discuss with you the what are the solutions. So the first question is, which of the following is a low resolution structural biology technique? So as we discussed, this is the slide I have taken from the last class discussion. So we discussed that in structural biology, we have three techniques which can give you higher resolution. They are X-ray crystallography, NMR and cryo-EM. But there are other techniques like circular dichroism, fluorescence spectroscopy, electron paramagnetic resonance, small angle X-ray diffraction. They will not give you a three-dimensional high resolution image. So from here, some small angle X-ray diffraction I had already written in the slide, and the answer here will be your option, this option A. Okay. So wherever you are facing any doubt, you can ask me. Then what is the doubling time for E. coli? So this is very easy. So the doubling time for E. coli is 20 minutes. So you know, you should actually have this knowledge by heart. Then only you can actually answer this question. So I have also listed various doubling times or generation time for other bacteria as well here. So the doubling time for E. coli is 20 minutes. So the answer is B. Then purification of proteins using NINTA column is an example of. So as I discussed last class, NINTA is by the affinity of this histag to this Ni nickel NTA column, right? So this is from the name itself, it is coming as affinity chromatography. So the answer to this, the means NINTA chromatography is an example of affinity chromatography that we again discussed in the last class, right? So I'm going in means discussing one question after another. If you have any doubts, you can stop me in between. Which of the following techniques separate the proteins on the basis of their size? So as we discussed, the mechanism of size exclusion chromatography, from the name itself, or the gel filtration chromatography, this is also known as size exclusion or gel filtration chromatography. So from the name itself, it is telling you that you are excluding some size, means on the basis of size you are actually separating your protein. So the answer to this question, fourth one, is C, that is gel filtration chromatography or size exclusion chromatography. Identify the equation stated below. So as we discussed today, the what is this equation? 2d sin theta equals to n lambda. This is your famous Bragg's equation, that is the B is the correct answer. Now again we have 5 more questions in the week 3 assignments. So in the above 5 means questions, if you have any doubt you can ask me. Otherwise I can go to the next 5 questions. Okay. Okay, so if not, then okay. So in the following equation below, the term delta H is. So delta H is this enthalpy term. As we discussed today, the enthalpy term of the crystal formation is modestly favorable. Remember, it was written minus small, right? So modestly available, this one. So the favorable, right? Because of the formation of this hydrogen bonding. So the answer to the question here will be C. Sixth answer will be C. Then 0 degree of orientation and translations are found in. So this I need to discuss with you. Let me means give you a means in a nutshell. Okay, so seventh question I will discuss with you later. Okay, so hold for few minutes, because going into the symmetry you need little bit knowledge. Okay, when in the equation below, the term T delta S solvent for protein crystallization is. So T delta is solvent. For solvent, as I mentioned, it will be a huge gain of entropy. So it will be a largely favorable process. So it will be favorable process. So it is eighth answer will be A. Okay. Then which of the following can be used to differentiate between a protein and salt crystal? So what is the protein dye test? As I said, you can use different dyes for looking into that. You can crush the crystal and look into whether it is brittle or it is soft. You can dehydrate it. So all of this will be the correct answer. So D is the correct option here. The last question is identify the method of protein crystallization given in the image. As you see, the drop is hanging to this cover slip. So this is the hanging drop method that we discussed today. Okay, so the answer to this question is option C. So only seventh question is left. For that, what we can do, I can actually discuss with you a little bit more about this crystals. So we were here, this is the lattice. So you have heard that I am telling you lattice planes, right? So some questions should come to your mind. What is a lattice then? Lattice is anything is which is a three-dimensional, means if you arrange atoms in a three-dimensional space in some form, okay, so that is known as the lattice, right? So if you have a three-dimensional form or three-dimensional, that is a lattice is a set of points constructed by translating a single point in discrete steps by a set of basis vectors. So just like if you imagine the crystal, that you are translating the unit cell into x, y and z direction. For lattice, you imagine that you have a point, a single point in space, and you are translating it in x, y and z direction. For crystal it was a unit cell, because the unit cell is made up of lattices. Okay, so we are going inside unit cell. So to make a unit cell you have a single point of atom that is actually translating in x, y and z direction and forming a lattice. What are the properties of a lattice? In Euclidean space, lattices are in infinite array. Means you can put infinite array of points in the x direction, y direction and z direction. So that can be a lattice. Okay, but that is not real. That is an ideal scenario. Lattices have translational periodicity. Translational periodicity means that you can translate it into x, y and z directions. Means you can take this whole lattice and translate it into x, y and z direction. It will not change the form, or it will not change its form. This, if it is like this, if you translate by one unit cell, you will have the same lattice only. Okay, so that is known as, in x, y and z direction, if you translate, then you will have the same lattice only. So that is the translational periodicity. So how to construct one lattice? You can play a game where you put some points in. You can put a single point here and try to translate it with certain periodicity. Means in a distance. For example, in a distance, I am translating it in one direction. So it is going from here. Again from A, it is going from here. It is going from here to here. So everything have a distance of A. Okay, so this is periodic, but this is a 1D lattice because you have only one dimension here, right? But again, if you put these two, one this line, another similar line in another direction, that will actually, and try to again translate these points. This will create one plane. That is your lattice plane. That is the 2D lattice. Now if you imagine you have one axis coming towards you from this screen, and again you use this axis in that direction and try to translate those points, you will get a cube or a three-dimensional structure like this. So this is your 3D lattice. So 3D, 1D lattice have only one lattice parameter, that is your distance. 2D lattice, for constructing 2D lattice, you need to have this distance parameter and also an angle with which your two axes are aligned with one another, these two axes. So this is one angle and two distance you require to create a 2D lattice. For creating a 3D lattice, you require three angles of all these axes made by together, and also these unit vectors, that is this A, B and C. So six lattice parameters in a 3D lattice, three distance and three angles. Okay. Now, as I said, these lattices will make this unit cell. So this unit cell, this slide we already discussed a few moments earlier. Okay, and then how these unit cells are made? These lattices make certain crystal systems. Okay, what are the crystal systems? The way the means as I said, there are six parameters of a lattice, that is A, B, C and alpha, beta, gamma. On the basis of this difference in this A, B, C and alpha, beta, gamma, you have seven crystal systems. Means these lattices will have seven different shapes. But there are different also lattice types. What is lattice types? Lattice types give you the arrangement of the atoms in that lattice. If it is a primitive lattice means in the eight corners of a cube you have eight atoms. If it is a body-centered lattice, then apart from these eight atoms in the eight corners of a cube, you have an atom in the center of that cube. If it is a face centered, then in each and every face you have one atom, and as well as the eight atoms in the eight corners. Then again you have this C centered. So in the C centered you have what? You have eight atoms, and in two planes again you have two atoms present, right? So this is the C centered means if you have something centered, means C centered means that will be present, these two atoms will be present in between the AB plane. If it is a B centered, then the atoms, these two atoms will be present in between the B centered. If it is B centered, then it is present in between the AC plane. If it is A centered, then it is in between the BC plane. Okay. So this is the way the different centered lattices are present. These are the different lattice types. Now if you combine all this together, you get actually 14 various different lattices. 14 various lattices you get. Okay. So this is what you get here. Okay. So from these 7 crystal systems and lattice types. Okay. So then if someone asks you that what is the total contribution of this means each atom in these lattices, you can actually calculate from here. So in 8 corners you have 8 atoms, and those 8 atoms will be sharing with the other lattice which is adjacent to it. So each atom is contributing to that lattice how much? There are 8 total atoms and 1 in each corner. So in each corner the contribution is 1 by 8, but there are total 8 atoms. So that's why 8 multiplied by 1 by 8, that is 1 total atom should be present in a simple cubic lattice. If it is a face centered, then you have 8 atoms in the corner. That's why 8 multiplied by 1 by 8. But again 6 atoms in the faces. But in the face, if you imagine this is your face center, there is again another lattice present here. This atom is contributing half to this lattice and half to this lattice as well. That's why its contribution is half. So 6 multiplied by half, that is 3. 3 plus 1 is 4. So the total contribution is 4. If it is a body centered, then there is a single atom in the body, inside in the center of the body. That is this one. So this is a single atom. And other 8 atoms are present at 8 corners. So that is 8 multiplied by 1 by 8, plus, that is 1 plus 1, that is 2. End center, again there are 2 atoms, but they are contributing to a subsequent, so there is a half contribution. So 2 multiplied by half, that is 1. 8 multiplied by 1 by 8 is 1. 1 plus 1 is 2. So this is how the contributions of different lattices or different atoms in different lattices is calculated. So this is a thing that is being asked sometimes in your questions as well. Okay, so this is the logic behind that. Now we are coming to the symmetry. So why we are talking about these lattice points, these unit cells and these crystal systems, everything? Because whenever you will try to solve the structure of a crystal, you will see that the crystal is a huge. It has a lot of electron densities. If you take each electron densities individually and try to calculate that how it is, what is the coordinate of that electron densities, it will be confusing for you as well as the computer. You can never do that. Why we have crystals? Because crystals has a particular order, it has a particular pattern. So if we can solve a particular pattern, the coordinates of a particular pattern, and try to translate it in x, y and z direction, the way the crystal is formed, then we can actually use the initial coordinate of that unit cell to get the coordinate of all the atoms present in the crystal. So that's why we should know with what unit cell, with what Bravais lattice the crystal is made up of. Now, how to connect one unit cell to another? That is done with the help of the symmetry operators. As I discussed that everything in the crystal is in a order and this is repeating with one another. So there are different symmetry operators which is actually connecting that these objects in the crystal. Okay, if you know what is the symmetry present in that crystal, you can actually solve the structure of the crystal very easily. So there are different kinds of symmetry operators. One is this mirror operator, one is this rotation operator, one is the center of inversion, and another one is this light plane. What is mirror? Mirror means just the way you look into a mirror. So it is just the mirror projection, like if it is a x y z direction coordinate is present here, then if you have a mirror along the y-axis, then the coordinate here of your image will be x minus y minus z, right? Okay, so similarly if you rotate, and if it is a rotation like this object is rotated by some angle, then it is called as the rotation. So it is denoted by 1 2 3 4. So this is 360 by n. So n is your these numbers, this 1 2 3 4. So if it is one full rotation, then you have a complete rotation and you are coming to the same point. If it is a two-fold rotation, then you are moving 180 degree. If it is a three-fold rotation, then this is you are moving 120 degree. So you are actually connecting one atom or unit cell with another unit cell with this rotation operator. If it is a screw operation, then you are moving 180 degree fast, that is two-fold rotation you are making, and then you are moving in one direction. You are making one translation, okay, in another direction, that is your x direction. Okay, so that is your screw. How a screw looks like? Screw looks like this, right? So screw means you are rotating as well as translating, rotating as well as translating. So that is the screw operation. What is center of inversion? Center of inversion means you have a rotation axis, and perpendicular to this rotation axis you have a mirror. Means if you have one point here, you first rotate it in this region. It will, if it is x y z, then here it will become x minus y minus z, due to this means rotation, right? Because the x, no, not the y, the minus x y minus z, right? The y will be positive because you are rotating along the y axis. Now you are taking a perpendicular mirror here. So now what will be happening? It will come here. So this coordinate will be minus x minus y minus z. So this is a center of inversion. So if you try to connect this point with this point, you will get a center of inversion. So that is your center. Center of inversion means first you rotate, then you take the mirror image perpendicular to the rotation axis. So that is your center of inversion. Glide means first you need to rotate, then you have a glide plane along which you first rotate and translate. Means you rotate, so glide is similar to your what you have. So here it is, you have a mirror plane, then you rotate. So mirror and then translate, that is glide. Here it was only rotation and translate, that is screw. If it is a mirror and then translation, then it is a glide plane. So they combining all this, you have 32 point groups. For these are all known as point groups, and all these point groups aim these operators when combined together, they form these space groups. There are total 65 space groups for protein and 32 point groups for proteins. Why this is so, you can look into it. Why not more than this? Okay, so from here again I will continue in the next class, because I have done this in a very rush, because I needed to discuss with you this seventh question. But I, from here I will again start in the next class, because I know this is little bit tough to imagine and you need some time here. Okay, so let me discuss with you the week 7 seventh question first. So it is telling you 0 degrees of orientation and translations are found in. Okay, so 0 degrees and orientation. So protein in the crystal. So if you see, this is the unit cell which is actually translating. But when it is protein in the buff water, then it has all this translation and orientations, everything is present because of this entropy. Water is also, buffer is also the same case. But when it is in the crystal, the protein can itself do not move or orient. It is the inside the protein, this unit cell, they are arranged in such a way that we can relate one another with the help of these symmetry operators. So the answer to this question will be your option B. Okay, so this is what I needed to discuss with you today. Since this is 2 hours session is already been done, so we can actually leave today. And if you guys have any doubt, you can look into this video. And if you have any doubt, you can actually ask me in the next class. Okay, thank you. Bye bye.